Hey guys, Mr. P. In this video we're going to talk about how to draw the ultrastructure of a prokaryotic cell based on an electron micrograph. We're going to start by giving our cell a general outline, and that's going to be followed by the addition of the cell wall. So I like to start drawing the prokaryotic cell with a cell wall. We'll go ahead and label this cell wall. Next, we want to draw the membrane, and it is important to note that the cell membrane is on the inside of our cell wall, and the cell membrane is much thinner than the cell wall is. So we'll label that cell membrane. On the inside of our cell membrane are a variety of structures, the first being cytoplasm. Next, within the cytoplasm are a series of small organelles called ribosomes. Now these are specifically 70S ribosomes. (70S) ribosomes would be the size or the designation of ribosome in a prokaryotic cell. Next, prokaryotes will have a plasmid. It's a circular ring of DNA. So we'll label this plasmid. The last thing, at least internally speaking, will be the nucleoid region. Now this is a nucleus-like structure. It contains the DNA associated with a prokaryote, so we'll label this nucleoid region. And it is important to note that the DNA associated with the nucleoid region is without histones, and so we call that naked DNA. Okay, that is DNA without histones. The only other thing we need to include on the outside of this surface is a flagellum, which is a long whip-like tail that provides locomotion for this cell. And the last thing we need to include are these small kind of hair-like projections that extend from the cell wall out. They are on the outside of the cell wall. We call these pili, and these serve as attachment points for the cell and its environment. These would be all of the requirements for earning full marks on the prokaryotic cell. If you learned something in this video, give it a thumbs up. If you have a question, leave it in the comments. Consider subscribing, as it will truly help out the channel. See ya!